Hello! In this video I want to take you on a brief tour of the spinal cord using a very special data set that was kindly given to me by Evan Calabrese over in the US. This data set uses a post-mortem spinal cord which has been scanned in an MR scanner at incredibly high resolution, and in fact the resolution is so high that we're getting essentially histological grade images from this data set. So just to give you a bit of orientation about the screen that we're looking at, and to warn you that occasionally I'll be glancing upwards because I do have some other settings on a separate screen that I might need to change during this tutorial. But the screen that we've got here, we're showing a transverse section of the spinal cord on the top right, a coronal section in the middle panel, and at the bottom right a sagittal section. On the transverse section you can see the classic appearance of the spinal cord, where we've got the grey matter forming this central H or butterfly shaped region, surrounded by white matter which comes out as lower signal in this scan. On the coronal section we can see once again the white matter as this darker region on the coronal, and then we can see how the grey matter is organised in a longitudinal column running the length of the cord, and we can even spot the central canal of the spinal cord there in the middle. Furthermore, on both the transverse and the coronal planes we can also see that we have got some roots connecting to the spinal cord. This is the dorsal side at the top and the ventral side at the bottom, so these are presumably dorsal roots back here and ventral roots just here. At the bottom, in the sagittal plane, we can see the termination of the spinal cord at around this point here at the conus medullaris. We can see the white matter of the cord and the grey, and furthermore we can see how this more distal end of the cord is surrounded by the cauda equina, all of these dorsal and ventral roots forming this horse's tail shaped structure down at the bottom. On the left hand side in purple we have a 3D representation of this data set. So I've been able to put all these slices together and create a 3D rendering, and furthermore, just to help us to orientate ourselves, we've got the cut end of the spinal cord shown in the transverse plane just here, and I can actually move up and down through these sections to give us a different view and to show us where we have cut through the cord. So the first thing that I think we should do, and I'm just changing my view slightly, the first thing I think we should do is just take a look at the cord in the transverse plane and think about how it changes and relate that to this overview image. Now the first thing that you note when you look at the spinal cord is that its calibre is not uniform. We note that at the top end here, and particularly at the mid to lower cervical regions, we have an enlargement, and this is called the cervical enlargement, and we have a cervical enlargement because this correlates with the connection to the brachial plexus. So within this cervical enlargement we've got lots and lots of lower motor neurons and sensory neurons sending information out to the muscles and collecting information from all of the dermatomes of the upper limb. Then we note as we go down the cord becomes quite thin in the thoracic region and then starts to widen out a bit more to form the lumbosacral plexus at the lumbosacral enlargement at the bottom, which connects to the lumbosacral plexus. Now the lumbar enlargement is less prominent when we're looking at this three-dimensional view, and if I zoom in just a little we can try and find it. So the lumbar enlargement is at around about this point here. It's less prominent just because the whole thing is surrounded by the cauda equina. So let's go down through the sections and have a look at what the cord looks like in the transverse plane at the various levels. So let's look in the middle of the cervical enlargement to begin with. So I'm going to just scroll down and we'll make our way down to about the central widest portion of the cervical enlargement, and we can see here this is the middle of the cervical enlargement, probably around about C6, something like that, C7, and we can see that we've got lots and lots of white matter. All right, we've got lots of white matter because, of course, at the top of the cord, the motor tracts have barely yet distributed their fibres along the length of the cord to the body. And furthermore, at the top of the cord in the cervical region, all of the sensory pathways have converged together and are now all making their way up to the brain. So we've got lots of white matter here, and we can see that the grey matter, particularly the ventral horns, are very, very large. If we go down now to about the mid-thoracic level, we see that the cord becomes far smaller. Okay, so the cord is much, much smaller at the mid-thoracic level. However, we've still got a reasonable amount of white matter because that's carrying information up from the lumbar enlargement, and also it's yet to distribute motor information down to the lumbar regions. Within the thoracic cord and the upper lumbar cord, for that matter, remember, not only do we have a dorsal horn and a ventral horn, but we also see this lateral horn, which contains sympathetic preganglionic neurons. Then as we continue to descend, we start to see the cord getting bigger again, and as we get to the lumbar enlargement, we'll see the grey matter regions expanding, as we can see happening now. Just overshot slightly there. I'll go back up a little. Okay, so there we are. So here we are in the lumbar enlargement, and we can see now that the absolute amount of white matter has started to decrease, but we've got big regions of grey matter here corresponding to motor and sensory neurons, which are distributing out through the lumbosacral plexus. And furthermore, we can see that the chordate equina is forming nicely surrounding this lumbar enlargement. As we make our way right down to the bottom, we see that the cord tapers down to pretty much nothing, and remember that's at about the L1 vertebral level. So we're looking here at the lower sacral and even the coccygeal cord segments. Okay, and then the cord just disappears as the filum terminale down amongst the filaments of the chordate equina. So that's an outline of the different levels of the cord, just correlating it to our 3D model. Now, in the paper where this data was published, the workers actually quantified some parameters here. So what we'll do next is bring up a graph and just correlate that to what we see in the transverse sections. So here we've got a graph here on the left hand side showing some numbers relating to white and grey matter and also the dimensions of the cord. So we're starting right at the top here. Whoops, let's just bring that up again. I'll just reorganize my screen. Just bear with me. Okay, so starting right up at the top here, and I'll just show us the red section, which is the transverse plane. Starting right at the top of the cord, you know, here we're at the cervical enlargement here. And what we notice, if I take us to about the middle of the cervical enlargement, which is at around about this point here, about sort of 8 centimeters down from the top, we can see that the left-right dimension is quite high, and we can see that, can't we, on the cervical cord? It's laterally wide, all right? And furthermore, we can see that at around about this level, it has a high cross-sectional area. In fact, it's got the highest cross-sectional area of anywhere along the cord. And also we do see in the red line that there is a higher fraction of grey matter at this level, corresponding to the segments that feed into the brachial plexus. As we move down into the thoracic region now, so now we're in the thoracic spinal cord, we see that the left-right and the anterior-posterior dimensions are smaller, which we said before, that the thoracic cord is thin. And furthermore, the cross-sectional area is low. As we go down towards the lumbar enlargement, we see that the left, right and AP dimensions increase, as we can see around about here. And furthermore, that the amount of grey matter as a fraction does really start to increase as we get lower and lower and lower. Alright, and in fact when we get down to the sacral cord, down to around about this level here, we can see that there's hardly any white matter, and the vast majority of the cross-sectional area is grey matter that corresponds to this spike just here. So that's us taking in the spinal cord along its full length and looking at some specific parameters. Let's now take a look at some 3D reconstructions that I've made of the various regions of grey matter. And what we'll do is we'll start off looking at the grey matter of the cervical cord. And once again, this is where I just need to go to my other screen, and I'm going to just bring up a little 3D representation of the grey matter of the cervical cord. And the way that I did this was essentially I went through the slices and I just labelled, just painted on essentially the grey matter as I saw it. For your orientation, this is the ventral aspect or anterior with the ventral horns. This is the dorsal aspect or posterior with the dorsal horns. And you can see that we can move up and down with our section here that we can observe in the top right hand screen. Now there's a very important message here. And that message is that, you know, we're so used to looking at the spinal cord as a series of transverse sections that we forget that the grey matter and the white matter exist in 3 dimensions. These are not 2-dimensional structures, they are 3-dimensional columns of tissue. And if we zoom in, we can really appreciate that. So here is the ventral horn in the cervical cord, and we can see that it has a Z-axis as well, it has a length. Alright, and of course this ventral horn extends all the way down to the sacral and coccygeal levels even, and extends all the way up into the brainstem as the motor nuclei within the brainstem. We can see that the dorsal horn is just the same, and if we rotate this round, we can see that the dorsal horn forms a column within the spinal cord. So it is not only the white matter that is organised longitudinally, the grey matter is as well. Specifically to the cervical enlargement, we can see that the grey matter here is large, there's a big area of it as we discussed earlier, and particularly these ventral horns are very prominent to distribute through the myotomes in the brachial plexus. So that's the cervical grey matter. Let's take a look now at the thoracic grey matter. So now let's just change our view and go to the thoracic grey matter here. So I'm bringing the level of the section down. Okay, here we go. And we can now see that we're looking at the thoracic grey matter. Now if we zoom into the slice, we can see that we have the traditional dorsal and ventral horns. So here's the dorsal horn and the ventral horn. We can see that the grey matter as a proportion of the total cross-sectional area within the thoracic cord is relatively low due to the fact that the dermatomes and myotomes at the thoracic level are relatively small. And furthermore, we can see the lateral horn in the thoracic level. Now, once again, the lateral horn, like the dorsal and ventral horns, is a longitudinal column of neurons. And in fact, this has another name as well, known as the intermediolateral cell column. Intermediate because it's between dorsal and ventral horns, lateral because it's sticking out laterally. So this is the intermediolateral cell column, or the lateral horn of the spinal cord, containing sympathetic preganglionic neurons. And this is really nicely demonstrates how this is a 3-dimensional structure. So that's the thoracic cord. Let's take a look at the lumbar cord now. Now the lumbar cord is a bit I've prepared this in a slightly different way. So let me bring up the model of the lumbar cord. Oops. So I just need to switch off that and centralize this so we can rotate it. So what I've done here with the lumbar cord is I have rendered it semi-transparent. and you'll see why that's the case shortly. So let's just take our cross section down here like this, and we can just see how that correlates. So we can see in brown we've got the lumbar cord. And the lumbar cord is characterized by having a large amount of grey matter and very big very big dorsal and ventral horns, due to them distributing to large myotomes and dermatomes in the lower limbs. However, when we look at the grey matter, we discover that the grey matter itself is not homogeneous. Not only is it separated into dorsal and ventral horns, but also these dorsal and ventral horns themselves have subdivisions. And this has been known for many, many years. However, it was only really in the 20th century that the functional implications of this became fully understood. What I've done is I've not actually done this sort of entirely anatomically. I've kind of semi-guessed where these so-called cell columns are, but they are pretty accurate. In the 3D image, you can see we've got 4 columns of cells within the ventral horn of the cord, and I've labelled them in green, blue, yellow and red. And you can see that also on the transverse section, and I can maybe just fiddle a little bit with the orientation of our other sections, just to give us a view of how these sit in space. They're not coming across brilliantly on those, so I won't persist in that. So let's just focus on the transverse section. Within the ventral horn of all regions of the cord, we have groups of lower motor neurons which are organized according to their function. The medial-most groups of cells supply muscles which are themselves most medial in the body. This green column supplies the axial musculature. So, for example, the erector spinae is supplied by cells represented in the medial portion of the ventral horn. As we move laterally through the ventral horn, we supply successively more distal groups of muscles. So the blue column of cells here supplies muscles of the limb girdles. Of course, this is lumbosacral enlargement, so this would be muscles at the gluteal region, but if we're looking at the cervical cord, it would of course be pec and rotator cuff muscles, for example. So the blue cells supply girdle muscles. The yellow cells supply muscles which are intrinsic to the limbs, for example the vasti or rectus femoris, whilst the red cells supply muscles of the most distal portions of the limbs, such as the hand or the foot. And these are columns of cells which exist in 3 dimensions. And we can see that very, very nicely here. I've just segmented this out, and I've only focused on the one ventral horn, but of course it would be the same on the other side. So we can see that we have columns of cells, columns of lower motor neurons supplying different distinct regions of the limbs. This can be clinically important, but in my teaching I don't linger on that too much. But I think the main message is to realise that the grey matter of the cord is not homogeneous. There are distinct cell populations, and these exist in 3 dimensions. So that's a look at the grey matter in more detail, particularly from a functional point of view. I want to end just by looking at something which is a bit interesting in this data set. So I'm going to turn off the segmentations that we've looked at. And what I want to do now is just focus your attention once again on the transverse view. And we're going to go up to around about the mid-thoracic level. It was interesting because when I was looking at these data, I looked at them after I read the paper that had been published in detail, and I thought there's something strange going on here at the mid-thoracic level, because what we see is that the grey matter and to a certain extent the white matter have been distorted. So I'm just moving up and down here in approximately the mid-thoracic level, and you can see that really we've lost the symmetry. Symmetry is a very useful thing to be looking for when you're looking at imaging, because if symmetry has been broken, that can suggest that something pathological is occurring. So we can see it's very clear that the grey matter at this approximately T6 chord level has been disrupted. Now, it wasn't only this kind of imaging which was done in this study. What they also did was they did diffusion imaging as well, looking at the trajectories of the axons within the spinal cord. And so what we might be able to do is to see some clues, some evidence of damage, if we look at the trajectory of the axons at this region. So let's bring up now the axons demonstrated by using diffusion techniques. And essentially what this does is it looks at the diffusion of water, follows the diffusion of water within the spinal cord, and that of course should correlate with the trajectory of axons if water diffuses along axons by capillary action. So here, this looks like a solid block of red, doesn't it? But actually, if I zoom in, you can appreciate that it's made up of thousands of filaments, and really this is only a small sample of the axons within the cord. This is not all of them by a long shot. All right. Now if we go to for example up here towards the cervical enlargement and zoom in, what you note is that the axons are running mostly in the longitudinal direction, and we know that, don't we? We know that in the cord we've got ascending and descending pathways, and these are running through the white matter. If we look at the transverse section though, we do see that there are axons in the grey matter. Of course there are axons in the grey matter, because the cell bodies need to communicate with the white matter. Now this is the level at which that lesion is. So let's put on the section onto our image here, and let's try and find where that lesion that we found is on the longitudinal diffusion data. So here we can see, we know that the grey matter has been disrupted at this point, and if we zoom in we can see the region, the white matter around the lesion, and look, something very interesting is obvious here. The axons in this region do not seem to be moving up and down in an orderly fashion, all running in the same direction. We can see that they seem to be going in the left-right direction, and this is picked up very nicely using the diffusion data. Some of them even seem to be looping around, doing strange things. And this is actually quite typical from a neuropathological point of view of a spinal cord injury. This patient, however, did not have any clinical history of spinal cord lesions, so it's really quite interesting, and it seems that there were no clinical features of any cord lesion at all in this patient. So we don't know whether how this was caused, and in fact there is even a possibility that this could be caused by post-mortem artifact, maybe some damage that occurred during removal of the cord from the body, although there was nothing at all documented to suggest that. So this is just a very interesting aside, demonstrating the beauty of these diffusion imaging data sets, and what they can demonstrate about both the normal cord and the pathological cord as well. So thank you very much for listening. I hope you found this interesting and useful in your studies of the spinal cord, and a big thanks from me to the workers who provided this data. Thank you.